Welcome back. Today, we're diving into the future of medicine with CRISPR technology, a tool that's transforming genetic disorder treatment. Imagine a world without sickle cell anemia, cystic fibrosis, or Huntington's disease. This isn't science fiction, it's the potential of CRISPR. Join me as we explore 7 diseases that CRISPR could potentially cure and the hope it holds for millions worldwide. Sickle cell anemia affects millions globally, causing significant health challenges and impacting the quality of life for those who suffer from it. This genetic disorder is particularly prevalent in regions such as sub-Saharan Africa, India, and the Middle East, but it also affects individuals worldwide. The root cause of sickle cell anemia is a single mutation in the hemoglobin gene. This tiny change in the DNA sequence has profound effects on the body. This mutation leads to the production of abnormal hemoglobin, which deforms red blood cells into a sickle shape. These deformed cells can block blood flow, leading to severe pain, known as sickle cell crises, and causing damage to organs over time. The impact on life expectancy is significant, with many patients facing a reduced lifespan. The burden of managing this chronic condition is immense, affecting not only the patients but also their families and healthcare systems. However, recent advancements in genetic research offer new hope. CRISPR, a revolutionary gene editing technology, has the potential to precisely target and correct the mutated gene responsible for sickle cell anemia. CRISPR works by using a guide RNA to locate the specific DNA sequence that needs to be edited. Once the target is found, the Cas9 enzyme acts like molecular scissors, cutting the DNA at the precise location. This precise cutting allows scientists to either disable the faulty gene or introduce a corrected version, effectively addressing the root cause of the disease. Clinical trials have shown promising results. Some patients who have undergone CRISPR-based gene therapy have experienced fewer pain crises and a significant improvement in their overall quality of life. Despite these advancements, challenges remain. Optimizing the delivery of the gene editing components to the right cells and ensuring the long-term safety and efficacy of the treatment are critical areas of ongoing research. The possibility of a cure for sickle cell anemia is now within reach, thanks to the groundbreaking work being done with CRISPR technology. This could transform the lives of millions of people around the world. Imagine a future where sickle cell anemia is no longer a debilitating condition. A future where children born with this genetic disorder can live full, healthy lives without the constant threat of pain and organ damage. That's the promise of CRISPR. This revolutionary technology holds the potential to not only treat, but possibly cure genetic disorders like sickle cell anemia, reshaping lives one cell at a time. Cystic fibrosis is a genetic disorder caused by mutations in the CFTR gene, leading to the production of thick, sticky mucus that builds up in the lungs and other vital organs. This excessive mucus clogs the airways, making it incredibly difficult to breathe and significantly increasing the risk of severe lung infections. Over the years, medical advancements have improved the life expectancy of those with cystic fibrosis, but it still remains lower than the average population. Enter CRISPR, a groundbreaking gene editing technology that has the potential to correct the CFTR mutation at its source, thereby restoring the production of functional proteins. Early studies conducted in lab-grown cells and animal models have shown promising results, indicating that CRISPR could effectively target and repair the defective gene. However, translating these promising findings into viable human treatments requires extensive further research and clinical trials to ensure safety and efficacy. The potential of CRISPR to transform lives is undeniable, offering a beacon of hope for those affected by cystic fibrosis. Imagine a future where CRISPR not only clears the airways but also improves digestion and overall quality of life for individuals with cystic fibrosis. The ultimate hope is that CRISPR could one day offer a complete cure, eradicating the disease at its genetic root and allowing individuals to lead healthier, longer lives. For families affected by cystic fibrosis, this technology represents a monumental shift, providing a glimpse into a future where the disease no longer dictates their lives. Researchers around the world are tirelessly working to make this vision a reality, driven by the promise of CRISPR and the profound impact it could have. With each step forward, the hope grows stronger, and the dream of a world without cystic fibrosis becomes more tangible. As CRISPR technology continues to advance, The possibilities for treating and potentially curing genetic disorders like cystic fibrosis are expanding, bringing us closer to a future where breathing easier is not just a hope, but a reality. Huntington's disease is a devastating neurodegenerative disorder caused by a mutation in the HTT gene. This genetic mutation leads to the production of an abnormal protein that progressively damages brain cells, resulting in severe motor dysfunction, cognitive decline, and psychiatric symptoms. The impact on patients and their families is profound, as the disease gradually robs individuals of their ability to think, move, and function independently. The abnormal protein, known as mutant huntingtin, accumulates in the brain, causing widespread neuronal damage. Over time, this leads to the loss of brain cells, particularly in regions responsible for movement and coordination. The relentless progression of the disease means that symptoms worsen over time, leading to a complete loss of independence and ultimately death. However, there is hope on the horizon. CRISPR, a revolutionary gene editing technology, offers the potential to target and disable the mutated HTT gene. By precisely editing the genetic code, CRISPR can reduce the production of the toxic protein, potentially halting or even reversing the damage caused by the disease. In animal studies, CRISPR has shown promising results. Researchers have successfully reduced levels of the toxic protein in the brains of mice, leading to significant improvements in motor function and overall health. These findings provide a glimmer of hope that similar results could be achieved in humans. Translating these promising results to human patients, however, presents a complex set of challenges. Ensuring the safety and efficacy of CRISPR-based therapies in humans is paramount. Rigorous clinical trials are necessary to determine the best methods for delivering the gene editing tools to the brain and to monitor for any potential side effects. Despite these challenges, the potential of CRISPR to slow or even cure Huntington's disease is immense. If successful, this approach could not only transform the lives of those affected by Huntington's, but also pave the way for similar treatments for other neurodegenerative disorders. Imagine a future where Huntington's disease is no longer a death sentence. A future where individuals can live full, healthy lives without the fear of this devastating condition. This vision is within reach thanks to the groundbreaking work being done in the field of gene therapy. Researchers around the world are pushing the boundaries of science to make this vision a reality. In state-of-the-art laboratories, scientists are tirelessly working to refine CRISPR technology and develop safe, effective treatments for Huntington's disease. The dream of a cure drives their relentless pursuit. Their dedication and passion are the driving forces behind the progress being made. With continued research and support, the hope of a future without Huntington's disease becomes more tangible every day. Muscular dystrophy causes progressive muscle weakness due to gene mutations. Duchenne muscular dystrophy, the most common form, affects young boys, often cutting lives short. CRISPR offers hope by correcting gene mutations responsible for muscle degeneration. Animal studies show CRISPR can improve muscle function and increase lifespan. Delivering CRISPR to all muscles in the body is a challenge. Despite this, the potential for CRISPR to slow or cure muscular dystrophy is exciting. Imagine children with muscular dystrophy living full, active lives. Researchers are dedicated to developing CRISPR-based therapies. The dream of a cure fuels their relentless pursuit. Hemophilia disrupts blood clotting, making minor injuries life-threatening. Current treatments involve regular infusions of clotting factors. CRISPR offers a potential cure by correcting faulty genes responsible for the disorder. Imagine a future where hemophilia patients no longer fear bleeding. CRISPR could enable the body to produce its own clotting factors. Researchers are exploring various CRISPR-based strategies. One approach involves correcting genes in a patient's stem cells and transplanting them back. These cells could produce healthy clotting factors, potentially curing the disorder. Challenges remain, but progress is encouraging. The possibility of a cure brings hope for a brighter future. Thalassemia disrupts hemoglobin production, causing fatigue and weakness. Current treatments involve regular blood transfusions. CRISPR offers a revolutionary approach by targeting genetic defects. Imagine a future where thalassemia patients no longer need transfusions. Researchers are exploring CRISPR-based strategies to correct faulty genes in blood stem cells. These cells could produce normal hemoglobin, potentially curing the disorder. Translating findings into human treatments requires further research. the advancements in CRISPR research offer hope for a healthier future. The dream of a cure drives relentless pursuit. Retinitis pigmentosa, RP, causes progressive vision loss, often leading to blindness. CRISPR offers hope by targeting gene mutations responsible for RP. Animal studies show CRISPR can restore light-sensitive cell function in the retina. Translating these findings into human treatments is challenging. The potential of CRISPR to restore vision is immense. Imagine individuals with RP regaining their sight. Researchers are dedicated to developing CRISPR-based therapies. The dream of a cure drives their relentless pursuit. The hope is that CRISPR could one day restore vision and independence. CRISPR technology offers hope for cures once considered unimaginable. From sickle cell anemia to retinitis pigmentosa, CRISPR has the potential to transform lives. If you found this video informative, don't forget to like, comment, and subscribe. Share your thoughts on which disease CRISPR will cure first.